Today's new concept under the unit number 4 plant breeding, that is the male sterility and its significance. First of all, we'll see the definition of male sterility and then study the important features of male sterility. The definition of male sterility is the absence of functional male gametes, or the male sterility is characterized by non-functional pollen grains while the female gametes functionally normal. The male sterility occurs in nature due to mutation. The important features of male sterility are: it is an important outbreeding device which prevents autogamy and permits allogamy. That means prevents self-pollination and allows cross-pollination. The second important feature, that is the presence of male sterility, leads to heterozygosity in species. Third important feature is the male sterility is caused due to pollen or anther abortion. It results from the action of nuclear genes or cytoplasmic genes or both. It occurs in nature through spontaneous mutation as well as can be induced artificially by chemical or physical mutagens. The male sterility can be observed in all diploid species of crop plants, both in a wild as well as in a cultivated. Male sterility, it is classified into three groups, that is the genetic male sterility, cytoplasmic male sterility, and cytoplasmic genetic male sterility. First of all, we will see the genetic male sterility. It is controlled by a single recessive gene, that is the small ms. The exceptions in a safflower, the male sterility, it is controlled by dominant genes. The male sterile alleles arises spontaneously or may be induced artificially. The male sterile lines may be maintained by crossing it with a heterozygous male fertile plants. The significance of that genetic male sterility, it is used in hybrid seed production. Then second, that is the destroying of male fertile plants from the female lines is costly. As a result, the cost of hybrid seed is higher. To overcome this problem, the male sterility has been utilized commercially in USA, successfully used in a castor. In India, it is used in a seed production of Kajanas kajan, cotton, sunflower, as well as in a cucurbits. Then second, that is the cytoplasmic male sterility. This type of sterility is determined by the cytoplasm of zygote comes primarily from the egg cell. The progeny of such male sterile plants would always male sterile. It is known in many plants like maize, Nicotiana tabacum, sorghum, bicolor, Helianthus anus, Oryza sativa, as well as in a Brassica gentiana. The significance of that cytoplasmic male sterility, it is used for the producing the hybrid seeds in a certain ornamental plants or in a species, whereas the vegetative part is of economic value. Then third type, that is the cytoplasmic genetic male sterility. When the cytoplasmic male sterility is expressed in a male sterile line which is known to possess a nuclear gene, that is the capital R, for restoring the fertility of pollen, it is called as cytoplasmic genetic male sterility. The expressions depends upon the nature of nuclear genotype with respect to restorer genes. The plants would be a male sterile in presence of males. The cytoplasm only if the nuclear genotype is a small r, that means the pure recessive, while the restorer gene is the dominant in condition whether in a capital R in a capital small r suppresses the effect of cytoplasmic male sterile genes. Thus, the expression of male sterility depends upon an interactions between the nuclear and extranuclear genes. The significance of that cytoplasmic male sterility is it is used to produce hybrid seeds in a maize, bajra, cotton, and jawar. Thank you.